DNA replication, RNA transcription, and protein synthesis. What is DNA replication? DNA replication is the process by which DNA makes a copy of itself during cell division. The first step in the DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary base of the DNA together, A with T, C with G. The separation of the two single strands of DNA creates a Y shape called a replication fork. The two separated strands will act as a template for making new strands of DNA. One of the strands is orientated in the 3 to 5 direction. This is the leading strand. The other strand is orientated the 5 to 3 direction. This is the lagging strand. As a result of their different orientation, the two strands are replicated differently. Leading strand. A short piece of RNA called a primer comes along and binds to the end of the leading strand. The primer acts as a starting point for DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide base A, C, G, and T to the strand of DNA in the 5 to 3 direction. This sort of replication is called continuous. Lagging strand. Numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Ozaki fragments are then added to the lagging strand, also in the 5 to 3 direction. This type of replication is called the discontinuous, as the Ozaki fragments will need to be joined up later. Once all of the bases are matched up, A with T, C with G, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet another complementary nucleotide. The new strand is proofread to make sure there are no mistakes in the new DNA sequence. Finally, an enzyme called the ligase seals up the sequence of DNA into the two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotide. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule, half is brand new. Following the replication, the new DNA automatically winds up into a double helix. RNA transcription. Transcription is the first step of the gene expression. During this process, the DNA sequence of a gene is copied into the RNA. Before transcription can take place, the DNA double helix must unwind near the gene that is getting transcribed. The region of opened up DNA is called transcription bubble. Transcription uses one of two exposed DNA strands as a template. This strand is called the template strand. The RNA product is complementary to the template strand and is almost identical to the other DNA strand called the non-template strand or the coding strand. However, there is one important difference. In the newly made RNA of RNA, all of the T nucleotides are replaced with U nucleotides. The site on the DNA from which the first RNA nucleotide is transcribed is called the initiation site. Nucleotides that come from the initiation site are given negative numbers and said to be upstream. Nucleotides that came after the initiation site are marked with positive numbers and said to be downstream. If the genes that transcribe encodes a protein, which many of the genes do, the RNA molecule will be read to make a protein in a process called translation. RNA polymerases are enzymes that transcribe DNA into RNA using a DNA template. RNA and polymerases build a new RNA molecule through base pairing. For instance, if there is a G in the DNA template, RNA polymerases will add a C to the new growing DNA strand. RNA polymerases always builds a new RNA strand in the 5 to 3 direction. This is, it can only add RNA nucleotides A, U, C, or G to the 3 end of the strand. RNA polymerases are large enzymes with multiple subunits, even the simple organisms with bacteria, humans, and other eukaryotes are 3 different kinds of RNA polymerases 1, 2, and 3. Each one specializes in transcribing certain classes of genes. Plants have an additional 2 kinds of RNA polymerases 5 and 4, which are involved in the synthesis of certain small RNAs. To begin transcribing, a gene's RNA polymerase binds the DNA of the gene at the region called a promoter. Basically, the promoter tells the polymerase where to sit down in the DNA and begin transcribing. Each gene, or in bacteria, each group of genes and transcribed together, has its own promoter. A promoter contains DNA sequences that let RNA polymerases or its helpers protein attach to the DNA. Once the transcription bubble has formed, the polymerases can start transcribing. What is protein synthesis? Protein synthesis is the process in which polypeptide chains are formed from coded combinations of single amino acids inside the cell. The synthesis of new polypeptides required a coded sequence, enzymes and messenger, ribosomal and transformal ribonucleic acids, RNAs. Protein synthesis takes place within the nucleus and ribosomes of a cell and regulated by DNA and RNA. Protein synthesis steps. Firstly, decoded for a protein, a chain of amino acids in a specific order must be copied from genetic information contained within a cell's DNA. The initial protein synthesis step is known as transcription. Transcription produces an exact copy of a section of DNA. This copy is known as messenger RNA, which must then be transported outside of the cell nucleus before the next step of protein synthesis can begin. The second protein synthesis step is translation. Translation occurs within a cell organelle called ribosome. Messenger RNA makes its way to and connects with the ribosome under the influence of the ribosomal RNA enzymes. Transfer RNA is a molecule that carries a single amino acid and coded sequence that acts like a key. This key fits into a specific sequence of 3 codes on the RNA, bringing the correct amino acid into place. Each set of 3 RNA nitrogenous bases is called a codon. To make the copies stretch of code transcription, we need enzymes called